AP 120, Lab 10, topics blood cells and antibodies. So here is a slide of a blood sample. And blood, as we know, is our liquid connective tissue. And it has three big categories of cells within it. It has the erythrocytes, also known as the red blood cells, lots and lots of those. Platelets, little teeny tiny guys, and then the leukocytes, and the leukocytes, as we know, have various subtypes. So erythrocytes, again, most common blood cell is the erythrocytes. They are reddish in color, they have a biconcave shape, it sort of looks like they're little donuts, kind of, and their function is to carry oxygen. Then we have platelets. Platelets are little tiny structures, much smaller than red blood cells, and platelets help in clot formation, so very important for helping to stop blood loss. Then we have the lymphocytes. Here is a lymphocyte. Lymphocytes are important for our specific immunity, so immunity attacking specific pathogens. They have a very large nucleus that fills a lot of the cell and a sort of light blue cytoplasm, and they're either the same size or bigger than the red blood cells. And about 25 to 33% of all of our leukocytes, all of our white blood cells, are lymphocytes. So here is a lymphocyte. And then monocytes, we have two monocytes on this slide. Monocytes are large cells, much bigger than red blood cells, and they engage in phagocytosis in tissues. So in the bloodstream, they are inactive. They're only active in tissues. Then we have neutrophils. Here's a lovely neutrophil. It engages in phagocytosis, and it is active both in the bloodstream and in the tissues. Its nucleus has lots of little blebs, as it were, 2 to 5 little lobes. And the neutrophil is the most common leukocyte. So over half of the white blood cells are neutrophils. So the most common blood cell is erythrocytes. The most common leukocyte is the neutrophil. Here is the basophil. Again, very easy to see with all these dark purple blobs everywhere. And its job is to increase inflammation, and for people with allergies, it helps cause the allergic reaction. And then finally, the eosinophils. Eosinophils, with their multi-lobe nucleus and their reddish granules, eosinophils function to fight parasites such as worms, and also to limit inflammation. And those are the various cells found in a sample of blood. So when you look at blood, you can spin it down really fast in a centrifuge, and you see that about 55% of it is blood plasma and about 45% are the erythrocytes. And the percentage of blood volume that is erythrocytes, that's the hematocrit. So around 45% is the normal hematocrit. Anemia is a condition where you have a reduced oxygen carrying capacity in the blood. This can be because you have few erythrocytes, so less than 40%, or you don't have enough hemoglobin per erythrocyte, or you don't have enough iron. So these are three different things that could lead to anemia. Polycythemia is the opposite. It's when you have too many erythrocytes. So when you have greater than 55% of the blood volume is red blood cells, then that is also a problem. Our specific immunity, this is our ability to fight a specific pathogen, to focus on destroying that specific thing. That is controlled by and primarily a function of the lymphocytes. There are a bunch of different kinds of lymphocytes, and they are involved in mounting an intense immune response against a single pathogen. So they will target one pathogen and ignore other pathogens until other lymphocytes in the community say, hey, wait a minute, I know that one. So we have helper T lymphocytes. Helper T lymphocytes, they turn on the specific immune response. So we need them to activate to recognize the pathogen. Oh, it's influenza. And then to say, hey guys, we need you to get active so we can fight the influenza virus. One type of lymphocyte they activate is the cytotoxic T lymphocytes. Cytotoxic T lymphocytes will then find our own body cells that are infected with influenza and destroy them. So cytotoxic T lymphocytes destroy our own cells that are infected by that specific pathogen. And then the helper T cells also activate B lymphocytes. And the B lymphocytes will then turn into plasma cells, and plasma cells produce antibodies against the pathogen. So in our example, the helper T lymphocyte activated by influenza will turn on the B lymphocytes that specifically will become plasma cells that produce antibodies against influenza. So only the B lymphocytes against influenza get activated, and they produce antibodies that will bind to influenza and help to deactivate it and lead to its destruction. So antigens, what are antigens? They are just structures on the surface of a cell or a virus. So they're just something that has a shape that's on the surface, something that our immune response can recognize. Because again, our white blood cells cannot see. So what they do to recognize cells, they go around and touch them. So they go around and touch their own cells and say, oh, look, these are shapes that I expect. And they touch, say, a bacteria, and they go, wait a minute, here's a shape I do not expect. That is not the correct shape for this body. I'm going to generate an immune response. So again, antigens are just structures that have a shape that are on the surface of the cell or the virus. Antibodies are special proteins that bind to that specific antigen. So we had helper T cells make contact with this antigen, go, wait, this doesn't belong here. They activate the B cells that become plasma cells that produced antibodies for this specific antigen. And when the antibodies bind to it, it can cause a lot of different possible immune responses. But the key thing being is to inactivate and or destroy the pathogen. And agglutination is when a bunch of antibodies cause clumping because they bind to multiple bacteria, multiple viruses, multiple cells, and cause a clump. Now, if something's really tiny and it gets clumped, that's a good thing because then it's easier for, say, the neutrophils to recognize it and then destroy it. But when it's huge things like, say, red blood cells that clump, then those big clots can block blood vessels. Here's a little video about B lymphocytes. Antibodies. Well, as we said, antibodies are proteins. They're actually a protein complex, to be more exact, and they are a type of immunoglobulin. They have this core Y shape to them. Some of them have multiple Ys, and they are made by plasma cells, which again were originally B lymphocytes. And so the ends, the short ends of the Ys are the regions that vary from antibody to antibody, and these are the regions that bind to different kinds of antigens. So this antibody here only binds to the yellow antigen. Why do we care? Well, antibodies are important for our specific immune response, and times when it's really important to get it right is with vaccines, blood transfusions, and organ transplant rejection. So vaccines are a way for us to avoid getting sick, but to be able to generate lovely antibodies against that pathogen so that we hopefully don't get sick should we ever get infected. So basically, a lot of vaccines now will inject the antigen into your bloodstream. Your B cells react, produces lots of antibodies against that antigen. And then there's also memory cells produced. And then if you're infected in the future, you're able to destroy the pathogen before it makes you sick. With blood transfusions, as we know, you get the wrong kind of blood transfused in, then that can cause a major agglutination effect and cause you to die. And then organ transplants, obviously, we don't want the person receiving the organ to have their immune response attack the new organ, because then it's not going to be able to function. And that is it for this part of Lab 10.